is the current tiger location. And the Bengal tigers are primarily found in 4 different countries: India, Bhutan, Bangladesh, and Nepal. India, of course, has the highest number of Bengal tigers, and Nepal probably has the second highest, although we don't have the exact number for Bangladesh because they haven't done the survey yet. So what are the conservation issues and challenges threatening tigers? If we tease those, loss is primarily the main reason that we are losing tigers, because there is no prey, there is no food. And also, oftentimes, and then we know through constant interaction with communities around national parks, Nepal shares probably same tigers as India. They go back and forth. They don't obey the geopolitical boundaries. So these are the main challenges of conservation, or their dwindling numbers. So back in 2009, there was this big conference on Kathmandu Global Tiger Workshop. And they realized that saving wild tigers is important. Nepal Tiger Genome Project is working in that space so that we provide information that is needed for effective tiger conservation in Nepal and beyond. So what was the goal of Nepal Tiger Genome Project? Well, the goal was building wildlife genetic capacity for Bengal tiger conservation in Nepal. And the objectives were to enhance capacity, to apply molecular tools for wildlife conservation research in Nepal, and to create spatial genetic database for wild Bengal tigers in Nepal. Started 2011, ended 2013, and we focused on Terai Arc landscape. So what we focused on was using genetics. What we also focused on was perhaps getting the genetic information through non-invasive method. So we focused on tiger feces, or scats as they call it. Our team systematically went through the whole of Terai Arc. Each grid that you see over there is 15 kilometers by 15 kilometers. And there were the exact grids that were laid for the previous rounds of camera trap surveys. So we wanted to correlate really well with the previous round of camera trap surveys and see how genetic methods compare to the camera traps. So this, we hoped, gave a consistency in terms of survey. 108 grids, 4 different national parks, all possible corridors, and areas 50% or more likelihood of tiger presence. We went out, we covered more than 24,000 square kilometers, 30 different teams, collected over 1,200 samples in 2 seasons. It was a monumental effort on our part, and we are very thankful and appreciative of the Department of National Park, WWF, and NTNC who helped us achieve this. Just give me a second. So we went out in the field. A lot of the things that we did in Nepal Tiger Genome Project, we had a lab before, but the lab was not comprehensive enough to do all things in Nepal. So with the help of USAID and through the project, we acquired a genetic sequencer that allowed us to do DNA fingerprinting. But we also have to train our lab personnel to build their skills to do this. And so what we ended up doing was doing everything from the scratch. in a very, very systematic way that wasn't done anywhere else. So in a lot of different ways, we were pioneer in terms of developing the protocol. We were pioneer in terms of trying different things out in Nepal by Nepalese. Of course, we had help from our experts, our collaborators from the US, but a lot of this was done here in Nepal by Nepalese. And so we are very proud of that. What you are seeing over here is the scat feces of the tiger, a potential tiger. We developed a method where we can preserve it very nicely. We can track it very nicely. And then we captured all the field data. Not only we captured data about the scat and all that, but we also captured information on the habitat. We also captured information about their positioning. So all of this we try to put together, and which I will try to show later. So we brought that in our lab source of our genetic material. So we bring it in our lab, we get DNA out of it. So we do the extraction, DNA extraction, and we have a library of this DNA from, we ended up doing only for 770 samples because we didn't have resources to do 1,200. We didn't actually expect to collect 1,200 samples. So we ended up doing 770. The process is you extract the DNA, and then we run it through a genetic test that allows us to tell whether it is tiger or not, and that's species identification. And then if it is tiger, then we can tell whether it's male or a female. That's sex identification. And once we have found that to be a tiger, then we can do DNA fingerprint to separate between two different samples, whether they are coming from one individual. It is called polymerase chain reaction, where we amplify a certain segment of the tiger DNA that are very specific to tiger. And what you see on this line is, let's say, 490 that you see, a band. If the band is there, then it's tiger. 503, no band, means no tiger. So we trained our field people extensively in collecting these samples, preserving these samples, tracking these samples, bringing them accurate, identifying the scat out in the field. So the margin of error, if you just rely on your visual, is 50%. On the sex identification, again, we looked for X and Y chromosomes. If it is a male, it will have X and Y. If it is female, it will have only X. And so in Chitwan National Park, we found 155 of the scat that were of male, and 84 of them were of female. Now, on the DNA fingerprinting part, we had to actually develop these markers, they call it, because no one has done it in a way that would be really effective. There are certain teams in India who have done it, pockets of them. They don't work together, but we actually talked to them and we said, when you did it, how did it look? Can you share the data with us? And so Dr. S.P. Boyle from WII was helpful in terms of giving us information about the markers they used with their tiger samples. We included those same markers in our panel because they were making this genetic database for their Bengal tigers in their triad, and we wanted to kind of have some kind of comparable data. Well, the estimation was 125 back then. So we still need to polish the methodology, the way we do it and all that. But the two techniques, in my opinion, correlate fairly well. And especially if you look at Bardia National Park and Suklakanta Wildlife Reserve, our estimation in Bardia is 59. The latest camera trap data from 2012 13 was about 50. And for Suklapata, our estimate is about 25. I think it's pretty close to the camera trap data that we have gotten so far. So, in conclusion on this, the camera trap data and genetic data, there's not much variability in terms of population census. Survey can't do is the genetic health of these populations. So if you look at Chitwan National Park, we were expecting about, so the term for this is heterozygosity, right? So we were expecting that Chitwan National Park was about 47%. So is that good or bad? Well, that really depends. In line of conservation, if you have 60% heterozygosity, then you have a good, healthy population. The other thing that we did was, well, if you have these tiger populations from 3 different national parks, if we all put them together and analyze it, genetically, do they separate out? Turns out, they do. So if you look at the Chitwan National Park, all blue, it's kind of clustering pretty well. And if you look at Bardia, that's clustering pretty well. If you look at Suklapata, that's clustering pretty well. Except for a couple of individuals. If you see, there is a Suklapata gene somewhere there. Migration, I don't know, or somehow that got transferred. There's a very, very limited gene flow. But in conclusion, on this part, whether we have a gene flow between metapopulation or subpopulation within the national parks of Nepal, we don't. It's distinct subpopulation in Chitwan, Bardia, and Suklapata. Is this worrying from conservation point of view? Perhaps. You know, we're thinking about doubling the tiger population in 2020. If we double it, are we also, you know, creating problems doing so, or is it feasible to double the population within the restricted area, within the current? So Chitwan National Park has almost 10,000 square kilometers. Bardia has 10,000 square kilometers. A lot less population in Bardia than Chitwan. So this is something that conservationists, policymakers, program implementers need to think about in terms of what would be the most effective way of planning so that we get double the number of tigers in these national parks. Is there a possibility of opening up a corridor linking Bardia with Chitwan, linking Suklapata with Bardia? Something to think about. Then what we did was, well, this is all good and all, but we want a very simple interface that can be related, or that can be, you know, understood by our program managers, a policy maker. So we know of those 5 samples in blue, they all came from same individual. And then we have the GPS location for these scats. With this, on GIS maps, and you can play with different things like habitat, man-made barrier, natural barrier, and you get these maps where you can look at, and I'll demonstrate to you the map that we have, where everything is visual. The interface is simple, right? Nepal, Chitwan National Park, GIS layering, man-made barrier, ecological information, habitat information. Each points are tigers. That we're very that. So let me demonstrate to you the geospatial map we have created out of this. Chitwan National Park, each dot there is verify tiger sample. Now, what we can do, we can go to each dot and click it, and there you go. The sample ID was this. It was a male, and there is the DNA fingerprint information. For a lot of people, it doesn't make sense. This makes sense. So that's why we developed this. And no one has done it for tigers anywhere else that I know of to get this kind of interface. So with this, we can figure out their territory, right, where they might have moved by this way. The other interesting thing, which might be of very interest for our Department of National Park folks and also some program inventors, conservation program, 2 tigers. This is the buffer zone in the Chitwan National Park. We also identified 4 common leopard. Here are the human settlements. And see how close they are. Some are as close as 1 kilometer away. And we talked about human-wildlife conflict a lot, right? And we can't always blame the tigers for it because we are encroaching their habitat. If we are encroaching their habitat, of course, we are going to run across problems. And this is an evidence that shows the high likelihood of human-wildlife conflict because the human settlements are so close to wildlife. Perhaps now we can think in terms of how to redesign our buffer zone so that this doesn't happen. The other interesting thing, and I think this is a very hopeful slide, after 1995 they were relocated. And we surveyed that area. We found tigers in there. There is a possibility that by restoring habitat, the tigers might come back. Very positive slide information. The other thing that we are kind of dabbling on and trying to do is there is a satellite that the US government has up for about 30 years now. They've been taking pictures of all parts of the world. What we can do is we can tailor it, so that's a Chitwan National Park. It's called Landsat. And what we can do is see how Chitwan National Park was 30 years ago, how it was 20 years ago, how is it now, and how it might look if things are all the same, business as usual, 5 years from now. Will we be able to keep Chitwan National Park as Chitwan National Park, or will we be seeing it smaller, having less of a biodiversity? Something to think about. With GIS, satellite maps, and other maps. And then we can say, well, tigers at this point really enjoy this kind of habitat. If they enjoy this kind of habitat, if we extend it, if we extrapolate out, what are the areas that could be a good habitat for them? So perhaps if we are thinking about expanding our national parks,